So far we've covered neurons, but these are not the only type of cells that occur in our nervous system. We also have glial cells that support neurons in many different ways. Another feature of the nervous system is the blood-brain barrier, which is important for regulating which chemicals can enter the brain. In this section of the lecture, we'll look at how glia and the blood-brain barrier support the functioning of our neurons. Glia literally means glue in Latin. Glia are sometimes also known as glial cells, neuroglia, or neural glue. For many decades, it was believed that glia didn't serve any major purpose in the nervous system. They were purely the glue that held neurons in place, hence the name. It was also believed that we had more glia than neurons in our nervous system. However, more recent research in the last 5 or 10 years has shown that glia play a vital role in neural functioning. Furthermore, these recent findings suggest that while glia are more numerous in the cerebral cortex, other areas of the brain, including the cerebellum, actually have more neurons than glia. There are multiple types of glia, including astrocytes, also known as star cells because of their shape, microglia, oligodendrocytes, and Schwann cells. Each of these types of glia perform slightly different functions within the nervous system. Astrocytes, also known as star cells because of their shape, provide physical support to neurons in the nervous system, hence the name nerve glue. They also play an important role in providing nutrition to neurons. Specifically, astrocytes take glucose and break it down into lactate, and this lactate can be used by mitochondria to power neurons. Astrocytes also store glycogen, which can be broken down into glucose in emergencies. Astrocytes also perform a housekeeping role by cleaning up dead neurons. They do this using a process called phagocytosis, which is where astrocytes engulf and digest waste products, a little bit like how an octopus covers and engulfs its food. Astrocytes regulate the chemical composition of fluids outside of neurons. For example, they take up neurotransmitters that have been released into the synaptic cleft, and they use these to synchronize related neurons, and they regulate things such as breathing patterns between neural pathways. Another type of glia is microglia. As the name suggests, microglia are the smallest type of glial cell. Microglia account for about 10 to 15% of glial cells in our brain. Microglia play an important role in our immune response within the brain. When an injury or pathogens are detected in neurons, microglia rapidly move to the lesion area to fight off invaders. Microglia communicate with other cells within the immune system and help reduce inflammation in neurons. Microglia also clean up after damage has occurred within neurons. That is, dead neurons, plaques, or tangles are removed using phagocytosis. Unfortunately, during this cleanup process, there is some collateral damage to neurons, and so microglia have been implicated in disorders such as Alzheimer's disease. Oligodendrocytes are another type of glial cell. An oligodendrocyte is my favorite word in all of biopsychology. Oligodendrocytes form the myelin sheath within the central nervous system. They do this by wrapping fatty lipid insulating tubing around the axon, which you can see in this diagram in blue. As the oligodendrocytes wrap their paddles, or what we call processes, around the axons, there are small gaps micromillimeters in width between these bits of myelin. These gaps are the nodes of Ranvier, and these unmyelinated parts of the axon allow fast transmission of signals along the axon as the signals jump from node to node. Schwann cells are pretty much the same as oligodendrocytes in their function of producing myelin. However, Schwann cells are only found within the peripheral nervous system. Unlike glia that live in the central nervous system and produce scar tissue, Schwann cells in the peripheral nervous system guide sprouting axons and allow regeneration of cells. You can see the Schwann cells wrapping around the axon in the image on the right. But notice that unlike oligodendrocytes in the central nervous system, which have multiple paddles, Schwann cells here only have a single paddle or process that wraps around the axon. I've just mentioned how microglia help fight off pathogens in the brain. but we also have another line of defense called the blood-brain barrier. Our body is exposed to many harmful and toxic substances in our environment. The brain is highly susceptible to damage from these chemicals, so it's really important that toxic chemicals don't ever reach the brain in the first place. This is where the blood-brain barrier comes in. The blood-brain barrier is a semi-permeable barrier between the blood and the brain. It's semi-permeable, which means that some substances can pass into the brain through the walls of the capillaries, while other substances cannot. As you can see on the left side of the figure here, in all of our blood vessels outside of the brain, the cell walls are loosely packed and substances can cross through the gaps easily. Therefore, they're permeable. However, if you look at the right side of the image, you can see that the capillaries in the brain do not have these gaps and the cells of the blood vessels are tightly packed together. As only some molecules will be able to pass through these gaps, the blood-brain barrier is semi-permeable, just like a strainer letting some things through and preventing others. Whilst the blood vessels are tightly packed in the brain, the blood-brain barrier is not the same strength throughout the entire brain and nervous system. For example, the area postrema, which is located in the medulla at the base of the brain, has a much weaker blood-brain barrier than other parts of the brain. Why is this? Well, the area postrema controls vomiting. So having a weak barrier here means that the brain can detect poisons more easily and get rid of the toxins by initiating vomiting, hopefully preventing these toxins from entering other areas of the brain and causing damage. So at this stage, you're probably wondering what substances can cross the blood-brain barrier into the brain and what substances are prevented from entering. As you can see in the purple arrows in this diagram, glucose, which provides energy to the brain, and amino acids, which are used to produce proteins in the brain, can cross the blood-brain barrier. This movement occurs via active transport, and so energy in the form of ATP is needed to push glucose and amino acids through the barrier into the brain. Other molecules can cross the barrier passively. That is, they don't require ATP or energy. They can just float through the barrier. And we'll talk about passive transport a little bit more in a while. Molecules that can cross the barrier passively include small uncharged molecules such as oxygen and carbon dioxide, and also molecules that are fat-soluble, that is, molecules that can dissolve in fat. These molecules that can cross are indicated by the green arrows in the diagram. However, you'll see that charged molecules and large molecules that can't fit through the gaps are not able to cross the blood-brain barrier. These are indicated by the blue arrows. This means that many molecules and a large number of chemicals can't get through to the brain. So with this in mind, how are drugs able to get into the brain and affect our nervous system and behaviour? Well, most psychiatric drugs have been designed in ways that they can be dissolved in fat. Because fats can cross the blood-brain barrier, these drugs are able to enter the brain. Heroin and nicotine are examples of drugs that dissolve in fats and can pass into the brain. However, one of the biggest challenges in psychopharmacology is figuring out how to develop drugs that can cross this blood-brain barrier. The treatment of Parkinson's disease is a really good example of this issue. We know that Parkinson's is related to depleted levels of dopamine neurotransmitter in the brain. Wouldn't it be great if we could just give someone a dopamine pill to restore this imbalance of dopamine? Unfortunately though, dopamine doesn't cross the blood-brain barrier, so we can't give people a magic dopamine pill. However, pharmacologists have discovered a clever way to get around this problem. They're able to give Parkinson's patients a drug called L-DOPA. L-DOPA is a molecule that can cross the blood-brain barrier. Once inside the brain, L-DOPA molecules can be catalyzed by enzymes into dopamine. And because they can create dopamine, this reduces the symptoms of Parkinson's. We'll go into more detail about this in the next topic on neurotransmitters and psychopharmacology.